Our keynote speaker tonight is someone who I'm a big fan of, Erika Augustine, who I can't remember when I first met her, but she has done some impressive work in the area of rare diseases and clinical trials. Erika is at the University of Rochester, where she did a combined fellowship in pediatric movement disorders and clinical trials design. And she has done some very wonderful work in pediatric movement disorders, and especially in Batten's disease. And I hope that you guys will welcome her to give us a little bit of some insight into her experiences and ideas. I'm not too tired at the end of travel, dinner, lectures, and all sorts of things, but I catch everyone on the beginning end of all the lectures, so hopefully there's still good energy. And thank you, Lori, for the introduction. And actually, the topic is Lori's idea as well, so thank you, Lori, for that also. So what I'd like to do today is talk a little bit about rare diseases and the current therapeutic landscape, and to talk about that in a historical context, spend a little bit of time talking about what's happening now and potentially where we'll go. And I realize not everyone is studying rare diseases, but hopefully there are some concepts that you can take away in terms of thinking about your own respective field, where your field has been, and how that might inform the work that you plan to do. So some of you may recognize this. It's a picture of a room, and the picture doesn't quite do it justice. But this idea of infinite possibilities, and where sometimes life and science meet art, came about during an AAM meeting. And so in 2018, 17, 18, I forget which exactly, the meeting was in LA. And I stayed for the whole week, which as you know, is a long week. And there came a day where I couldn't go to any more meetings. I couldn't learn anything more, and I just needed some air, and I wanted to go do something else. So I went to the Road Museum. And they had an exhibit. They had two exhibits of work, of an installation by Lipofuscinosis. And there were two. One was a room, and one was actually more like a box. Has anyone actually been to this installation? So a few of you remember. And when you go into the room, and this is the picture of that room, the souls millions of light years away, you step onto a platform and everything kind of melts away. And there are thousands of strategically placed mirrors and lights, and the room just sort of extends infinitely. And the same happens with this box. It's truly a box, a hole in it, and you put your head inside, and it's like everything around you disappears. And again, I think probably because I've been spending all that time thinking about science and the work that we do, there just seemed to be an immediate parallel of these infinite possibilities in terms of what's happening in the rare disease space, where we can go with therapy development, and really what may be possible for some of these currently untreatable fatal disorders. We're really embarking on a new era of neurology, and I think it's a really exciting time, and I hope that all of you are excited about the work that you're doing as well. So let's get to rare diseases. These are really critically important diseases, and I often say that they're the diseases of the child neurologist. Half of rare diseases impact children or start in childhood. More than half have neurological manifestations. They're a critically important cause of deaths in the first year of life. And when you start to look at the numbers, we generally define rare diseases as conditions impacting fewer than 200,000 people. That's our US-based definition. Other countries have similar kinds of prevalence-based definitions. And when you take those disorders collectively, there are probably more than 7,000 rare diseases affecting 30 million people in the United States and more than 300 million people worldwide. And so again, they really collectively are common and they share similar challenges. So it does make sense to start to think about rare diseases not only individually, but again, by these common features and common impact. Financially, they also have substantial impact. There was a study in Australia where perhaps medical conditions are accounted for 52% by rare diseases, but account for almost 15% of hospital-based costs. So again, as financial and public health models, they're really quite important. So I want to take a look at what we might consider the past. And we're going to talk about past in loose terms. Past, present, and future will overlap just a little bit. And I'd like to talk about those in the context and framework of neuronal ceroid lipofuscinosis, or Batten disease, as Lori mentioned, where I spend a lot of time thinking about these disorders, and actually just came yesterday from the annual Batten Disease Support Research Association meeting. So when we look back, first to clarify a little bit what NCLs are, for those of you who are not thinking about them every day, they're really quite more common than one would think. They're one of the most prevalent causes of intellectual and neurological deterioration in childhood, or progressive disorders, dementias in childhood. And there's a lovely study from the UK, an ongoing work, where they tracked diagnoses of children with dementia. And they took a look over a 12-year period from the early 2000s onward, and they looked at the children who actually had a defined diagnosis. There are about 2,500 children that they looked at overall. About half of those kids actually had a clearly defined diagnosis, and you can see that NCLs are actually fairly similar. Typically, you might think of common disorders, you think about lipodystrophies, you've heard of lipofuscinosis, but often the NCLs are not necessarily what you think of, but really should be. And they repeated this study some years later, again, a little bit more current, and found similar things. Their methods were slightly different, but the sample was larger, and again, we still found NCL disorders at the top of that list. Right there with Rett syndromes, even Tay-Sachs, Krabbe, adrenal lipodystrophy, all conditions that you're probably more likely to think of than NCL, but again, really tied there with prevalence. So remember these as disorders that, again, are leading causes of progressive, genetically based deterioration in childhood. So the history aspect of things. So the first published account of Batten diseases was back in 1826, and this is actually the original text in Norwegian. A colleague of mine, Heather Adams, found the original text, had it translated by Drs. Hager and Anderson into English, and this is really a case series about a single family that had 4 affected boys who each, around the start of what we would now think of as elementary school, around 4 or 5 years of age, developed a progressive blindness, then dementia, epilepsy, and motor disorder that really looked like Parkinson's disease, and much like old texts tend to have. It was really a beautiful report, really well written, well described, as if you would go look at some of our more modern stroke literature again and find these really beautiful descriptions. Really sort of set out what we now think of as being NCLs, and this is our first account there. And fittingly, it was actually in the first edition of the Medical Society Journal in Norway called AIR. But then we don't call it Stengel's disease, we call it Batten disease. And so the Batten eponym, and there are many eponyms in this field, goes to Frederick Batten, who in the early 1900s, some 80 years later, had the first description that had pathology that went along with it. And so the eponym went to him. And then a series of others, Gold, Spielmeyer, Janksky, Belshowski, Cliffs, Haltier, and Santagori, described other forms, some with similar juvenile onset in the early elementary period, some with an infant-type onset, and again, in the case of Cliffs, with an adult-type disorder. Batten disease refers to the juvenile onset type. It also refers to all of these disorders together, and even though we're looking at a 150-year period here, it still wasn't quite clear, is this a single disorder? Is it a group of just a couple of disorders? But there were these unifying features. We now know that it is a group of disorders, but again, until very relatively recently, it really wasn't clear exactly how many. These disorders are characterized by some unifying features. Clinically, blindness, motor dysfunction, movement disorders, epilepsy, premature death, dementia are all common clinical symptoms, no matter the age of onset to a degree. There's selective and progressive neuronal loss, and then intracellular accumulation of autofluorescent storage material in the form of ceroid lipofuscin that on EM has certain patterns that, again, once thought went with specific diseases, but that's evolved over time. initial approaches to therapy, again, 150 years in plus, to the recognition of the disorder, I want to call your attention to a few things and what the initial approaches to therapy development or experimental therapeutics look like. So the first is thinking about the NCL type. You can see that most of these have a juvenile neuronal ceroid lipofuscinosis kind of abbreviation or description here, perhaps with one exception of a late infantile form. Again, that was the form that we recognized well. It really stood out from other kinds of progressive disorders in childhood, and those symptoms had a fairly clear and evident path. And so we thought that that was something that was notable and unified a particular group. In terms of intervention, it was not clear what caused this. Certainly there was an inflammatory component, big on antioxidants, and so there were many studies of antioxidants, all with an attempt at disease modification, with one exception, which was a study in the 1980s to look at Parkinsonism. And when it comes to the type of study, the sample size, the duration of follow-up, with the exception of this one study of Parkinsonism, we really had a whole host of open-label studies without any comparator, including without natural history comparator, with one exception here, this sample of 43. The studies were fairly small for the most part, and even some of these larger size studies, it's a little bit deceiving to look at all of them because some of the larger samples include the studies of the smaller samples as well. And all were fairly inconclusive and had really wide-ranging periods of follow-up, and were really based on the data that were available at the time. So you see very short periods of follow-up, half a year, all the way to 18-year retrospective case series. So we really had to enter the modern era. It wasn't exactly clear what we were studying, and the mechanisms of disease had yet to be understood. So this is where things started in terms of thinking about NCL disorders. We very commonly think about these approaches being similar for other rare diseases as well. And so you might envision the past as being a period where we described phenomenology, where we grouped characteristics, and we linked those characteristics to pathological findings, but again, very precise and specific etiology had not yet been determined. And therapeutic approaches emerged in the late 60s, 70s, 80s, but for many rare disorders, there wasn't much progress that had been made, and really kind of honing in on just a few different concepts and testing those over and over again. So where are we now? Well, present, again, we use this somewhat loosely, but thinking about that late 80s, early 90s period to the current time. And again, there's a wide range of thinking about the present, but a lot of the processes and a lot of the approach that we take to thinking about diagnosis and therapeutic discovery are unified during that period of time. On the whole, when you think about rare diseases, often people talk about this diagnostic odyssey, where it takes a long time to achieve diagnosis, or what we would consider today a specific diagnosis. Certainly, this average of 5 to 7 years is a commonly cited one. Hopefully, as we move towards the end of that present period, that's a shorter period of time. But even within that, this idea of misdiagnosis is common. And when I talk about a diagnosis, I mean an etiologic diagnosis, not a symptom-based diagnosis or description of the symptoms at hand. Something that's more specific to etiology. And even now, even in the current era, about half of patients actually don't achieve a specific diagnosis, and those who have a rare disease, again, a specific etiologic-based diagnosis. And this poses some challenges. It makes for low prevalence estimates. It makes for limited interest in these diseases. It makes it difficult to test on the compounds when you really haven't ascertained the full spectrum of disease, and you really don't have an understanding of the full burden of disease in the population as well. And so there are two advances, one technical and one policy, that I would say are in our present era that have really revolutionized how we think about rare diseases and how we've been able to shape and advance therapy development for rare diseases. So that first one, in terms of technical advancement, has a lot to do with genetic diagnosis, approach to genetic diagnosis, and an increasing link between genetics and rare neurological disorders, or even rare disorders more broadly. similar to that timeline, although here I have it starting in the 50s, really dates back to the 1800s with Mendel and this idea that there are heritable factors that can be passed down. But then again, more than 100 years later is when the actual structure of DNA was discovered, where the idea of genetic code was determined. It was in the 1970s that the first animal models based on genetic disease were developed. The ideas, more robust ideas for gene therapy came around in the 1990s. Microarray technology in 1995, and then next generation DNA sequencing in 2005. And when we think about the Genome Project, started in 1982 with a draft of the human genome in 2001, you think about that 30-year period or 20-year period that really was required just to get to the full human genome. And we think about today, what does that look like when we think about whole genome analysis? So that technical advancement is one that's critically important. And then around the same time, while all of this was happening in terms of technology advancement in diagnosis and identification of genetic diseases, came the Orphan Drug Act. And many of you are familiar with this. We talked about, again, low prevalence, low interest, inability for companies to really robustly develop products and then also make a profit. And so a policy approach was taken in 1983 into 1984 to really try to change that and to develop and drive incentives to be able to move from that one to sometimes no new treatments approved or submitted by pharmaceutical sponsors for rare disease indications to develop financial incentives that made it reasonable to actually even consider therapy development for rare diseases. And so those financial incentives included tax credits, 50% tax credits on clinical trial costs, and extended time on patent, waiver of user fees, which just that alone is at least worth a few $1 million. And that act, starting in 1983, again, around the time that these other technological advancements really helped to shape how we move forward. And what we see overall, not only thinking about rare disease, but thinking more broadly, that development of central nervous system disorders is on the rise. So if you take a look at the dotted blue line, certainly see a dramatic increase for oncology, but certainly a sizable increase over time, and the amount of development, the number of compounds in phase 1 to 3 studies or early to confirmatory studies over time on the rise, whereas many other disorders are relatively flat. And it's become clear that not only are there financial incentives for studying rare diseases, that there are hierarchies of success for rare diseases as well. So while we are all optimistic about our new ideas, I hope all of you are optimistic about what you are studying. Generally speaking, there's about a 1 in 10 chance that this will be successful, maybe a little bit lower than that for CNS indications. But when you take a look overall, and these go back to about 2015, so probably would be some updated, but about 10% of all diseases will have success in terms of a compound that enters phase 1, the likelihood of reaching approval is about 1 in 10. We're a little lower than that, around 8% for chronic, high prevalence diseases, but we're actually up at 25% for rare diseases. These are often mechanism-based, targeted therapies that really get at the heart of the underlying disorder in terms of the primary cause, primary etiology. And so not only are there financial incentives directly, but there also is an incentive to study rare disease based on that high occurrence of gene-based etiology. So the Orphan Drug Act, like 1 or 2 approvals per year back in the 80s and before, number of products here on the y-axis, you can see that there's been a dramatic rise in the number of products annually, and this rate of about 45 to 50 new approvals each year has maintained from 2014 up to the present. And just to take a snapshot of 1 year, there are at least, in any given year, there are at least half a dozen to a dozen new compounds that are relevant for neurology that fall under orphan drug approval. So again, this has been an incredibly effective policy, and both the technical advancement along with the policy approach has been really successful. But what about the characteristics of these disorders that are, or the drugs that are being created and developed? And you can see that there's been a shift over time in a few things. So one, in dark green we have biologics, and light green, small molecules. You can see over time there's been a shift towards development of more biologics, and actually now, towards the end of the 2010 decade, it's almost 50-50. When we look at the representation of oncology, cancer in dark blue, metabolism and endocrine in light blue, and all other conditions in this grayish blue here. But here you see there's an increasing representation of orphan drugs and oncologic conditions, certainly based on the increasing importance of genetic etiology and oncology. Rare genetic conditions are also represented here in dark brown, are on the rise in terms of their orphan drug approval characteristics. And increasingly we're seeing that these are targeted therapies as compared to untargeted therapies. So the very nature of those drugs and the compounds that we use have shifted. So to go back to this timeline of genetic discovery, take a look at that last edition here of next generation DNA sequencing back in 2005. That really has dramatically shaped things. So in the next gen sequencing era, we really moved from thinking about one disease at a time and approaching that diagnosis, or even small categories of disease, as you well know, to thinking about symptom-based panels or disease groups in terms of panels, as well as whole exome sequencing becoming our first line approach. And this has led to previously unknown disease associations. And when we take a look at what the knowledge has yielded in terms of unknown phenotypes, for which we actually know the molecular basis, we've more than doubled that just in a 6-year period, and that continues to rise exponentially. Think about every journal that you open, and it seems week by week there's a new disease that's discovered or a new association that is made, and certainly in oncology, similarly, this rise in the number of mutations and unique variants continues to exponentially increase. I think nowhere is that better seen than in epilepsy, where, again, sort of this one-at-a-time, family-based gene discovery approach certainly had some success in the 90s, but then once that 2005 sequencing era began, we've had an exponential rise in our identification, understanding of monogenic etiologies of epilepsy. So what does that look like for back diseases? Well, we thought, again, maybe there was one group, maybe there were 2 or 3, an infantile, late infantile, juvenile, and adult onset disease. But even this now is still incomplete. It becomes too incomplete for the slide. But we can see that there are actually at this point more than a dozen different forms of NCL, each with a unique genetic basis. Different kinds of proteins, some secreted enzymes, some transmembrane proteins, and those profiles on electron microscopy that we thought were unique to a single disorder, we actually realize now we can see in all different forms. So our pathologic basis or our agent onset basis for making a diagnosis really didn't work. And what we were studying in these early trials were likely to be lots of different disorders with different mechanisms as compared to actually single diseases. And you can see as you look at the gene and also at the agent onset, or what we might think of as phenotype, we have both phenotypic plotogeny and genetic heterogeneity illustrated here. So this understanding also has really changed our approach to how we classify NCLs based on genetic type, mutated gene, and then very specifically the age of onset. So rather than just talking about late infantile onset disease, we actually now might say that person has CLNZ disease, the late infantile form, and again, we've been able to be more precise with diagnosis, but also more precise with thinking about therapy development. So along with those discoveries, it led to better understanding the role of these mutated proteins in the function of the lysosome. And there have been a few areas where there have been clear, rational places to intervene and to think about therapy development, whether that's CNS-directed enzyme replacement, correction of genetic information either through gene therapy, gene editing, stop codon read-through approaches. Understanding the genetic basis of the disease has directly led to an expanded pipeline of compounds for testing. So I want to take a moment to talk about, again, that present period in back diseases and where we are in terms of therapy. There are at least a dozen different NCLs. There's one approved therapy, and that's for CLNZ disease. A historical description is for what we think is probably CLN3 disease. This is a younger onset disorder in its classic form, although there are wide-ranging kinds of symptoms. And in that early onset form, children present with early language delay, refractory epilepsy, developmental first plateau and then regression. And then there's a rapidly progressive period where there's dramatic loss of milestones, and again, that difficult to treat epilepsy becomes even worse. Movement disorder is then usually in the form of ataxia, and again, there's loss of speech. Then there's a very prolonged late stage disease where children are essentially in a vegetative state prior to a premature death in the second decade. So these are devastating disorders. I'd like to show you a little bit of what that might look like. So this video is of a child who's now passed away. His family's been very active in the Baton community and with raising awareness of this. And in this first part of the video, the child in question, Noah, is running around with his sisters. You might notice that there's a little bit of interning of the left foot. They're very similar. He keeps up with the
Whoa, you guys are silly.
No concerns there. So then 6 months later, shortly after that video, he had his first seizure. And just in this 6-month period, you can see that this boy who was running around can no longer run around anymore.
Are you getting your cards out?
His speech is slurred. It's slurred. He crawls, but with difficulty. He wears a helmet due to refractory epilepsy with drop attacks. And at this point does not yet have a clear diagnosis. And so then this next part of the video is 2 years after the first video. And so he's no longer able to speak, he's blind, doesn't sit independently, he has frequent ongoing seizures, and that is the course of typical CLNZ disease. So in terms of trying to think about how to put that image, that video, that progression to that child into numerical form, some of our colleagues developed a rating scale that's very simple. It's essentially 3 points, or 4-point scale in terms of motor function, with 3 being normal function, and no loss or regression at that point, and 0 being the most severe regression, and then the same parallel for language. And so to map out what a typical course for CLN2 might look like, you can see that early on, in those first couple of years of life, really function is acquired in a way that's typical for age, but then very quickly there's a dramatic regression. And there's a dramatic loss of function from that time of that first seizure to really just within a couple of years. So we're going to spend the next several days talking a lot about various trial designs. And when it comes to rare disease, there are some that do actually lend themselves to thinking about a natural history approach. When you think about disease that is diagnosed early, or has onset early, I should say, there's a rapidly progressive phenotype that is fairly uniform and very clear. And so a natural history approach is one that is reasonable. And that's exactly what they took. There had already been established a registry, and they had historical controls who had been monitored with the same kind of approach. And so when it came time to test this therapy, so cereid alpha is a recombinant enzyme replacement therapy that replaces the enzyme that is deficient or defective in CLN2 disease, administered into the ventricles or intracerebrovesicular every 2 weeks through an Amaya. In the course of what was originally a 2-year study, and then further extended, you can see that when it comes to changing that motor language score that I mentioned, loss of motor function, loss of language function, those who were treated with ceroid alpha had a clear separation from historical controls. And again, although a concurrent control would be ideal, they were certainly able to demonstrate this difference. And on the basis of this study, on the basis of this single study, this drug was approved in 2017. And so this is a picture or videos of a child that I care for who is 5 in the video on the left, and who is 6 in the video on the right. Haley is still independently ambulatory. She moves with reasonable coordination, although you see more of her certainly is a bit ataxic. And so there's a clear difference here in terms of trajectory, the common trajectory of one child compared to where this child is at this stage in time. Certainly this is not a complete rescue, this is a complex disease, and the approval is for prevention of loss of ambulation. I told you about blindness, about dementia, about loss of language, about movement disorders, but again, the label is a very specific one. So it raises interesting questions for rare diseases when you have a clear molecular basis for the disease. If you correct the molecular basis for that disease, how do you think about what the potential outcome should be, what the impact should be, and how that might vary based on age and intervention? But again, I think for NCL disorders, moving from therapies that were evaluated in heterogeneous groups with broad approaches down to a well-defined, precisely defined molecular basis and intervention in a defined cohort has really come a long way. Certainly, I think there's no success at this point in time greater than that for spinal muscular atrophy. Many of you are familiar with the SMA story and the recent successes in this area. Spinal muscular atrophy, not only being a common genetic condition within rare diseases, but the most prevalent cause of infant mortality. We may actually have a question mark for that now if there's a setting of early diagnosis. Will this remain to be a leading cause of infant mortality? And hopefully the answer is no. I think the data are yet to be seen on that, but I believe that is the case. With SMA, you see that there are severe early onset infantile forms, then again late infantile or childhood forms that are more attenuated in severity. And the SMA timeline very much mirrors the NCL timeline, with the original descriptions going back to the 1800s, gene identification coming in the late 90s, the same as the sort of emergence of, again, a lot of the NCL timelines as well, and gene identification. And then finally, just being able to start to really well describe the cohorts and classify them, but develop some initial standards of care and approaches to thinking about disease quantification. All of that came relatively recently. And that first treatment for spinal muscular atrophy was approved in 2017, nusinersen, antisense oligonucleotide that modulates alternative splicing methylation 2. And in this trial, with concurrent controls, here you see a waterfall plot with change in what is essentially a clinical rating scale for motor outcomes. And you can see the nusinersen group where higher scores were better, had improvement in that score, and controls had no change, if not worsening. It was relatively uncommon to have worsening in the nusinersen group. And again, on the basis of these data, along with improved survival data, form the basis of the approval. And then right on the heels of that approval, in 2017, we now know about the approval for gene replacement therapy for enzyme for SMA in 2019. And in this panel, we look at two cohorts. In this case, a natural history controlled study, and looking at event-free survival, event-free being time through, or events being death and 24-hour need for ventilation. And these markers here in green, this first one, 75% of a natural history cohort is event-free. At this second one, at around 10 and a half months, 50% event-free. Third one, 25% event-free at 13 and a half months. And at 20 months, only 8% event-free from either invasive ventilation or death. And you can see that the gene replacement therapy group has far exceeded all of those, with a number of them being event-free completely at the last trial visit, represented by the stars. And so, again, on the basis of these data, gene replacement therapy for SMA was approved in 2019 and represents only the second gene therapy approved in the U.S. for any condition. So I think I would characterize our present time as translating knowledge into treatment, whether that is based on for migraine, targeted therapies for migraine, for DBS, but in the case of genetic disorders, gene replacement therapy is representing ocular therapy for genetic forms of retinal dystrophy, exon skipping therapies, and others. So we're still describing phenomenology. We're still grouping based on characteristics. But rather than looking always at pathology, we're linked to genetically based diagnosis. And those changes have really accelerated the development of new therapies. And so what about our future? So there has been a dramatic rise after NCLs, a dramatic rise in interest in therapy development. And certainly having one successful product approval really does change the landscape for rare diseases. It's clear that there are a group of individuals who can coalesce around the diagnosis, around a therapy, enroll and successfully complete. To be able to demonstrate that really goes a long way in developing interest. But this came along with other things as well. We talked about regulatory incentives. I just came from very engaged family groups. But there are an increasing number of clinician networks and collaboration. Our group has worked quite a lot on CLNZ disease and developing natural history models, and understanding the genetic basis of the disease has led to the development of more robust preclinical models, some of which are actually naturally occurring models, but also, again, generated preclinical genetic models as well. So there has been a huge change just in the course of the last decade from the time that I sat in your seats at this trial, at this course, thinking about a different study, a different trial, to now where there's a robust pipeline with many compounds ready for testing. So the NCL pipeline has really evolved from small molecule therapies alone, but again, a growing number of small molecule therapies, a brief period of time where there were some stem cell trials in very small numbers, but to more of a growing effort towards CNS-directed enzyme replacement, which has the potential to impact a number of rare conditions, gene therapy, particularly with the advancement of AAV therapies and knowledge of AAV9 and its particular tropism for the CNS, as well as RNA and DNA modulating compounds along with anti-inflammatories. So there really is a complex spectrum within that pipeline ready for testing. So now, again, there are still a few that are missing through here. 10 years ago, the pipeline was relatively empty, and now ready to enter trials, or actually in trials already, are gene replacement therapies for CLN3 and CLN6 disease, close on their heels, CLN1, 5, and 7, and then enzyme replacements in development for CLN1 as well as small molecule approaches. And for CLN7 disease, one of the few examples of true personalized medicine has been in development for a single patient with CLN7 disease with a unique mutation amenable to an ASO approach who has a therapy program specifically developed for her and has really fostered and advanced a number of conversations with the FDA about what does that look like on a large-scale basis. And so I'd be happy to talk with others about that as well. But it's been a very, very interesting time in the NCL field. So what do our emerging therapeutic strategies look like? So those studies in the 70s and 80s were targeting this idea of disease modification, but I think the SMA studies have really shaped the way that we think about what disease modification might look like. If you think about progressive disorders following along this red arrow, where again, without treatment or instead of placebo, there's a continued progression. Symptomatic treatments, you may have some benefit for a period of time, but then again, the progression is right back to where you started. And then ideally, we think about disease modification where maybe we can slow down the progression of disease. In a perfect world, for a while we would think about being able to arrest disease. But I think our SMA studies are showing us some other concepts as well. So Elsa Shapiro, a neuropsychologist who's really spent a lot of time studying pediatric dementias, has developed these models of dementia and how they might emerge and how they emerge in different ways in childhood. If you think about a typical developmental trajectory being represented by this solid line here, there are going to be some children with dementia who do have progressive cognitive loss, but just sort of slowly separate out from the curve. There are going to be some who have an acute loss of skills, and then again sort of fall up the curve, but are separated from typically developing individuals. In the case of intellectual disability, you really never had a period where you were on that curve, and then there are some who will fall off and will continue to get worse, or actually that's represented here. But what you might envision, not only in terms of therapy strategies, if they're early enough, that you may not just halt disease where things are, but that dementia occurs on a background and ongoing development in the childhood setting. So you may actually, if you halt disease, you may allow for some period of normal development or some development, and that seems to be what is taking place may not only be able to slow or ideally halt disease, you may actually be able to see some improvement when there's very early intervention and some room there for some typical development. And I would say in this future era, one of the ways that we're learning a lot in this genetic era to go forward for future questions. So our classic teachings about genetics, one gene, one protein, clear genotype-phenotype correlations. We think about trisomy 21 and Down syndrome. But what we see now is actually quite a lot more in the terms of genetic heterogeneity, where many genes can cause a single phenotype. Again, many NCLs can result in a later retail phenotype. Or there may be one genotype or one gene that different mutations result in different phenotypes. And so ATP1A3 mutations might be an example of that, where we see alternating hemiplegia, rapid onset dystonia, Parkinsonism, and now an increasing number of other kinds of phenotypes as well. With ATP1A3, the actual mutation phenotype correlations are reasonably good, but there are other conditions where that's not so clear. So CLNZ disease, for instance, can cause a early onset childhood phenotype that I showed you. It also is responsible for SCAR7, a recessive spinal cerebellar ataxia type 7 that occurs in the elderly. And whether or not if you create a molecularly based product, does that actually apply for all of the phenotypes at hand, or are there some differences that need to be teased out? And so these are some new questions. And in this era, it's been increasingly clear that there are links between rare and common disorders. I know that some of you study frontotemporal dementias, and so may be well aware of the connections between progranulin mutations or haploinsufficiency or progranulin mutations and one of the forms of NCL, which in homozygous form results in a classic NCL type, a fairly early onset NCL type. And in individuals with haploinsufficiency for progranulin mutations, you see retinal pathology and intracellular accumulation of ceroid diffusion, just as you do in NCLs. So are they truly related? Are there similar mechanisms? Can treatment and therapy be informed in that way? Between rare and common epilepsies, we see those kinds of links genetically. And then we also see these sorts of links between lysosomal storage disease and Parkinson's disease. Again, many of you are familiar with the links between Gaucher disease and links with Parkinson's disease, and that's one case in which that actually is being translated into a therapeutic approach. There is a company who is now targeting individuals with GBA mutations or risk for Gaucher disease with a specific compound, targeting that pathway, not in Gaucher disease, but for Parkinson's disease. And so there are ways in which there will be increasing numbers of personalized approaches to common disorders as well, based on rare disease. I think is yet to be seen what the success of those models will be, but certainly I expect to see more. There are some areas for caution. Although we've talked a lot about pediatric conditions and pediatric conditions in rare disease, when we look more globally at other kinds of policy intended to stimulate particular kinds of research, they haven't been so effective for pediatric approvals. And if you take a look on the right-hand side, looking at the number of compounds approved between 1999 and 2015 that actually have pediatric data available, let alone the pediatric indication or label, they're perhaps on the decline, and there are relatively few, perhaps better in recent years, but this is yet to be further evaluated. So I think when it comes to these barriers to therapy development, there are a lot of ways in which we've made advancement in terms of being able to get to more precise diagnosis. There's still those delays in those labs in terms of the threshold for early testing, the cost of testing, leading to inefficiencies there. There is a lot of work still to be done in terms of understanding the natural history, the variability across different populations, and a real lack of robust patient-relevant outcome measures. We will always have small sample size challenges, difficulties in recruitment, but these are some of the barriers to be tackled. And to go back to that disease example. So now we have a well-defined condition, or at least part of that condition is well-defined. There is a targeted therapy, and there is a clear indication for where the evidence is best. Some of the most important questions, like impact on lifespan, have yet to be answered. Whether or not there's the potential for impact on vision and epilepsy and other disease domains, again, is yet to be addressed. And as we saw in Pompe disease, when we had systemic enzyme replacement therapy, as children lived longer, we started to see emergence of other kinds of systemic pathology. So is that something that we'll see in CLNZ disease? There's already a hint that perhaps there's cardiac pathology that will emerge with age. The Ommaya reservoir was not intended for biweekly access for a lifetime. And so what does that mean in terms of long-term device durability? How do we optimize delivery? And certainly the approach, the treatment has been incomplete, helpful, but incomplete. And so what do those combination approaches look like? And how do we develop better methods that actually give us a sense of what the global disease burden might be? As we target for gene replacement therapy, gene editing approaches, skipping approaches, how do we think about targeting to overall levels of gene expression versus cell specificity? What's the most important factor there? What's the optimal route of administration for each particular disease? There really are quite a number of questions that emerge there. And that early intervention, what's the right timing for intervention is probably one of our main questions. How do we now have competing trials in small populations or in many small populations? And the SMA field, they handled that well. They were actually able to support multiple trials at the same time, and ideally other diseases will be able to do that as well. We still have these issues of timely diagnosis, looking for improved data quality, and then treatment access. And in the future of the Orphan Drug Act, I think are really quite pressing issues. So after that disease, after that approval, there certainly was excitement about the first approval both in the United States and in the EU of this groundbreaking treatment. First CNS-directed enzyme replacement therapy, first treatment for any NCL disorder, approved by the European Commission as well. But then the National Institute for Healthcare and Excellence in the UK said it's too costly. So this is a treatment that comes at a $750,000 drug alone price tag that is anticipated to be a lifetime kind of treatment. So how will we make sure that we have therapeutic breakthroughs that are beneficial and that we clearly understand the benefit, while at the same time put ourselves in a position where we actually think about worldwide application are all yet to be seen in this area. And the Orphan Drug Act and the future of the Orphan Drug Act, while it's been wildly successful, whether or not it's sustainable, again, is a very clear question. So our future is one where I anticipate we'll continue to advance and more rapidly advance the development of new therapies. We will have precise, meaningful diagnosis, but we have to change our concept of what we think of as treatment groups. We have 4 different phenotypes, 4 different types of expression of a single molecular entity. What does treatment look like across those groups? And these are things that we need to consider. And these connections between rare and common diseases also are things that we consider in therapy development. Early and multimodal intervention certainly will be part of that path forward as well. So there's lots of room for growth, but then also we need to slow down and think about our worldwide potential impacts. In the NCLs, although it's late, so to not talk about our data today, the work that we've been doing in NCLs has been very much focused on natural history. And so through a systematic approach, we've really been able to identify the specific symptoms involved in NCL disorders, understanding how they unfold and the sequence of onset of those symptoms. We've been able to put a quantification to certain types of disease progression, in this case physical progression, and been able to test some scientific questions of sex differences. Where we're going, we're trying to do a better job of modeling disease trajectory, identifying subgroups within this population, developing clinically relevant biomarkers in the form of anatomic evaluation. And then our final step is to really move more into some novel interventions and really look to develop new trials that should launch at the end of this year, which we're excited to look forward to. We talk a lot about these issues, and in September of this year, we'll be having a symposium at the University of Rochester focused on platform therapies, gene replacement, gene editing, ASO approaches, and both the cost and ethical and practical implications of these therapies. And so with that, we'll stop there and take questions.
trials for the rare diseases versus?
I think part of it has to do with, these days, the population being studied, and then also the therapy itself. So to take the example that I use for enzyme replacement, it's specifically targeting individuals with a classic form of CLN2 disease, with a genetic diagnosis, and then a therapy that replaces the enzyme that is deficient in that particular disease. And so if administration aside, distribution aside, as a concept, it's a therapy that should work. And many rare disease therapies in development these days really are there, getting at the heart of the primary molecular basis for the disorder. And so already from a conceptual standpoint have a higher likelihood of success. Take that in contrast to thinking about something like Alzheimer's disease or Parkinson's disease or even Tourette syndrome, which are likely many diseases with perhaps a final common pathway, but the interventions are actually downstream of that final common pathway. So uniform, group, part of the disorder, and I think that's why you're seeing those differences.
So one of the conundrums that we have is, that was a great talk, great overview. How do we deal with, from a clinical trials perspective, the issue of scalability? So you have 7,000 rare diseases, you talked about the genotype-phenotype correlation lab, where we get a lot of N of 1, N of 5, certainly not enough. You know, the statisticians have ways of doing this, and the FDA's been getting, I think, a little more flexible about this. But we're talking about 7,000. Forget about the treatment cost. Think about the cost we have to put out on the front end to do the research on them. So I'd love to hear your thoughts on that. How do we create something that's not just disease, you know, associates?
Well, I think that it's a major issue. It's both the upfront costs in development and the interest and time investment as well in a cohort of surfers who are actually willing to do that and who have the interest to identify the appropriate therapies. It's also an issue of thinking about natural history as well, as there are many, many new discoveries of unique genes and trying to think about how we approach understanding how diseases unfold in a rapid way, because as compared to the 50s, 60s, 70s, if not 1800s, where diseases were identified and there was hope for therapy to test at one point or another. It's now almost the opposite. There's been a gene identified, there's a compound ready for testing, but no one knows anything about the disease, and how do you accelerate that knowledge building process? So there's that piece of things too. And I think that one thing that comes to mind is trying to have how we build efficiencies into this approach. Some of the approaches, gene replacement therapy in particular, with the current administration, it's not as if we can have a series of N1 trials or a series of crossover studies. It's not an appropriate therapy intervention or not appropriate disease model for that either. But in terms of infrastructure, I think that we may learn a lot from our Mass General colleagues in terms of thinking about platform trials and to really have centralized approaches to bring together academia and industry in a way that is similar to what's happening in oncology. So that it's much more feasible, potentially still very costly, but potentially some ability to have some scalability for the more common rare diseases. You know, there's rare and there's rare. So there's 50 in the U.S. and there's 200,000 in the U.S. and all of that counts as rare. But I think we can start with the more prevalent rare diseases to think about some of these kinds of approaches.
I think that's a great response. Once you figure out a basic mechanism that will work, gene therapy, all the anti-nucleotides, things like that, you can apply it to a great many of those other diseases. And the gene therapy estimated, now we can try other conditions as well. The cure for, dystrophies, we have to try other conditions as well. These are the first gigantic steps forward that then lead to how do you attack all of this.
I think some of these issues are going to need a regulatory component to the solution as well. So thinking about gene replacement therapies and even just one step of oversight, the Recombinant DNA Advisory Committee, which just within the last year has been disbanded in favor of a new body, but largely because this body, which conducted an oversight review of every single gene therapy as it emerged, all of which were using very similar kinds of vectors, they almost never had any substantial advice to give. There were very few substantial protocol changes based on that RAC review. And so they've disbanded that in favor of a new body that's really looking at truly groundbreaking, innovative, not already in thousands of patients kinds of approaches that need that additional level of oversight. And then at the same time, thinking about ASO kind of approaches, you could envision trials where for a single disease, where there may be multiple ASOs for unique mutations, well, perhaps that could be a single trial, rather than a trial of one ASO for one unique set of mutations, and then that whole program, and then a new program, likely from the same organization, for another kind of indication. Just be able to do that in a more seamless way, and then similarly across disease phenotypes, not yet from an outcome standpoint address how we would do that. So go back to that CLN2 example. You have an infant with classic CLN2 disease, and you have an 80-year-old who has slowly progressive ataxia. How do you evaluate those individuals within the context of the same trial? What do the endpoints look like? What do the outcomes look like? To have some more nimble approaches to thinking about some of those within disease or within disease phenotypes may also be.
So I think like about 15 years ago, the whole concept of like buying a 3D printer on Amazon was basically probably inconceivable. Like that you build, like you just program a little 3D printer and you like build whatever you want, case for your phone. The iPhones were invented 15 years ago. I guess part of this looks like with this development pathway, all these molecules are just a little different, but then come with a price tag of be like $750,000 a year. I mean, it seems like somebody should design a 3D printer for molecules, right? Like so that people just they have a disease, the hospital buys the 3D printer for molecules, and you just print out the things that people need. Or maybe the government could fund it, like so that it's super expensive.
Well, I would say that in some ways I agree. I do think that that is part of the rapid advancement because particularly those last therapies that I've mentioned, they're all concepts where you can switch out a transgene or change out a component of the ASO, or it's a different enzyme replacement therapy that is then directed to the CNS. And so that's where there's this potential for that exponential part of the curve that seems to come. And again, I think that's where we are in those particular therapies as they're being developed. Some of the unanswered questions, though, there are many unanswered questions in terms of being able to have enough knowledge to understand optimal routes of delivery, exactly disease by disease, what particular cells need to be targeted, and can we target those precisely. Our preclinical models are limited in their ability to really model overexpression, and the models, and all of the platforms at the moment, are simplistic in their administration in that we're talking about pediatric disorders. Everything pediatric is developmental, including gene expression. And so the ability to modulate these therapies in a way that varies in a natural way that gene expression would, we're at our infancy in terms of that. So widespread 3D printers, not quite there yet, but I think there's a need to ramp up one and then.
First, great talk. It really just an elegant presentation altogether, and I think will also for the segues for commoners like me to weigh in on the discussion and ask a question. So the timeline from inconceivable 3D printers to 3D printers everywhere, and I can make my own gun in my basement if I it's fairly narrow. And so the commoner question is, from the multiple genes, one phenotype, one gene, multiple phenotypes perspective, these molecules are not exactly pure. And so what happens as we enter this exponential phase, and that person shows up in my ED at 2 o'clock in the morning with some described, not described complication of the molecule. I'm just curious, the question is more so, is there or are there some parameters around these molecule developments, and are there resources and places where one can go to say, you may or may not expect this, and is all that codified somewhere?
So it's growing and developing. I would say to look at CAR T-cell therapy, for instance, some of the neurological complications of that particular approach to oncologic treatment, some of those emerged in the context of the trials, but much of the knowledge that was gained came after. And I think that's going to be the case, where there will be knowledge based on the platform, so we'll gain AAV knowledge, ASO knowledge, gene editing knowledge. Then there will be disease-specific factors that have to do with the gene being targeted as well. And so there's a body of knowledge around AAV therapies, for instance, on hepatic inflammation, the time course of that, when to expect it, how to manage it, how that might vary based on route of administration. That's all just about the platform. It's about the kinks of the printer. But then in terms of the specifics of the transgene and overexpression or expression in undesired compartments, that will be more like CAR T, where it's going to be things that we learn after the fact, and we learn much down the road. So there are a lot of questions that emerge, and the kinds of complications that may be seen may be different very early in terms of intervention as compared to later.
And to piggyback slightly on that, he is not a common person. He's an amazing clinical trialist, but both of us are stroke doctors, and we're just kind of bitter because you get to shoot a molecule from like the 70s. It was proven in the clinical trial that came out when we were in high school in stroke patients in 2019. So we're bitter.
Many trials and the expectations are different. And so I think therapeutic misconception is far higher in rare disease populations than it is in controlled populations. It's on both sides.
So I just wanted to make a comment. I thought the CAR T cell is actually a great example to try to answer your question in a way. The fundamental problem is that we see each rare disease as a single disease, and we demand the same level of evidence to approve it. And the thing about CAR T therapy is that's not actually one therapy, because the cellular response is programmed for each patient. It's actually what's approved is a technique for creating a therapy. And what we need is enough examples of gene replacement therapies where the thing that's approved is the approach to the therapy and some level of testing, but not to prove that each therapy works for each disease. And there's a common often made in oncology is that the more we learn about cancer, the more we find out that everybody has a rare disease because no two people have the same cancer. And so I think the answer to the question is actually you made this comment, I'm just amplifying it. There's sort of a regulatory change in perspective where we're licensing strategies, not drugs and not molecules. And what we shoot for is not an assurance that every child with a rare disease receives a therapy that is effective, but a high fraction of them do. Just like we accept that when you get a drug for a disease, not everybody benefits from the drug, just hopefully a high fraction of those people. So the CAR T therapy actually provides the exact model for a change in regulatory framework. We just need a few more successful gene replacement therapies before we can establish the pattern. But if I was looking for a way to scale it, I'd be changing the approval request for being the process, not the molecule.
And there's some discussion about that in very early ways underway. Perhaps not around gene replacement, but possibly around ASOs. And thinking about approaches that have clear criteria for who candidate patients would be across various diseases and what level of preclinical evidence would be needed to move forward into human study for what is really maybe very individualized therapies where it's completely impractical to open a whole program that really would be this kind of approach where this is the framework and here are the guidelines and the boundaries within the work.
So one more thing before I know we're just keeping you up there forever, but this was a wonderful talk. And I also come back to the issue about outcome measures, which you touched on a couple of different times, and the development of outcome measures, and how difficult that is in these pleiotropic diseases, right? I see, I work with my shy in the CT clinic, I work with my tiny dystrophy clinics, and we're all thinking about different therapies. And the hardest part is having outcome measures that are going to be acceptable. I just wondered what your thoughts were on different pathways in maybe so far for both therapies. Health measures.
That too is not a solved problem. But I do think that there is room in a similar way to what I was just talking about in terms of moving away from every single disease has its own outcome measure, endpoint, clinical rating scale to thinking about groups of disorders and stepping back from that. From a pediatric standpoint, a lot of measures that were developed and validated in general pediatric populations, they don't work well for neurodegenerative diseases. The whole concept of having multimodal sensory impacts, blindness and dementia and motor dysfunction all together, it just doesn't work. But there are enough of those disorders where there's a sensory problem with dementia and motor dysfunction where it should seem tractable to develop new approaches to cognitive assessment or new approaches to motor assessment or even quality of life to think about very broad groups for development and validation of those measures. There's a lot of flexibility that has been discussed in terms of approaches to outcome assessment and then a real focus on functional outcomes. The challenge on some of that, though, is again, there's so much variability